So welcome back, everybody. We are beginning our third narrated presentation, third week of presentations, and this is on Chapter 3, which you can see is entitled The Molecules of Life. And this is going to cover all of those molecules that make up the remaining 30% of molecules that typically make up organisms that are not water, remembering that, of course, the average organism is about 70% water. It varies from organism to organism, but on average, it's around 70% water, which means that about 30% is non-water, and that 30% is pretty much built around skeletons of carbon. So one of the first things that we'll need to address in this chapter is why carbon, why not something else? And the answer to that is relatively simple. Then we'll look at what carbon is capable of doing, which is part of the answer to why carbon. And then we're going to take a look at the really large molecules, what we refer to as macromolecules, that make up this 30% of an organism that is built around these carbon skeletons, the non-water component of organisms. And along the way, we're going to discover some interesting things about the way these molecules take shape as a consequence of attaching things to carbon skeletons. And there are some ideas that we may dig a little bit deeper into than is presented in the textbook. We'll see how we're going. One of the things that concerns me about this chapter is the amount of information that's present, which means this presentation has a tendency to get a little bit long, but there are concept checks scattered throughout the presentation. It breaks up into a nice series of sections. None of them should be more than about 30 minutes long. So if you feel that you have hit information overload at the end of a section, feel free to put it on pause or to turn it off and to walk away and come back and take a look at it a little bit later in the day or on another day. Just remember to take a look at the full presentation at some point, because within the presentation, I will be covering everything that you need to know. And in terms of finding answers for exam questions, they're all going to be contained within this presentation. So if you really want to find out the answers to the exam questions and the sorts of things that you need to know, take a look at these presentations. That has always been the case. That will always be the case throughout the course of the semester. So without dragging out the introduction any longer, let's launch into a look at the molecules of life. And we begin by looking at something that is misnamed. And so what do I mean by misnamed when I say that? Well, you can see that the condition here is called lactose intolerance. And many of you probably actually suffer from this. You may know it or you may not know it, depending on how severe is your lactose intolerance. But what it means is that if you consume anything that is made from milk, so milk itself, ice cream, sour cream, yogurt, cheese, anything that has the milk sugar lactose in it causes you issues. And it's because you no longer produce an enzyme called lactase. So you replace the O in lactose with an A, and that gives you the name of the enzyme, lactase. And anyone who suffers from lactose intolerance actually has lost the ability to make the enzyme lactase. Now, when you are really young, you actually produce the enzyme lactase, which you are intended to do, because all mammals use milk as a source of nutrition for their young. But as the young develop teeth, which starts to happen relatively early in the development of all mammals, they have the ability now to start consuming normal adult food, solid food, and they move away from drinking milk. And for all mammals, with the exception of humans, The young get weaned off milk and start consuming solid food, adult food, and never return to consuming milk. And the exception, obviously, would be cats, but they're our domestic pets, and we actually provide them with milk to drink. Under normal circumstances, they wouldn't even consider milk as a source of food. It's only because we provide it for them. So humans are really the only mammals that continue to consume what we might refer to as baby food, milk, into adulthood. And in the big scheme of things, the intention would be that we would all lose the ability to make lactase and that we would all suffer from lactose intolerance. But to give it this condition a better name, we really ought to focus not on the 70% or so of people who actually lose the ability to make lactase, but we should focus on those that retain the ability to make lactase, about 30% of the population on average, because they're the ones that are unusual. The ability to continue to consume milk sugar and not suffer any digestive issues or metabolic issues as a consequence of that is the unusual thing amongst mammals. Because once you wean off milk and you start consuming solid food, you're not supposed to continue to consume milk. But humans have figured out ways to preserve milk in many ways. So the production of things like cottage cheese and yogurt and sour cream, anywhere where milk has been soured or fermented in very specific ways to produce product that we continue to consume, is actually a way of preserving milk without refrigeration. That's why it originally was produced, as a way of preserving the nutritional value of milk in the absence of refrigeration. And so in cultures that have practiced herding traditions and milking traditions, which is not universal across all human cultures. Not every human culture has a herding tradition, but in human cultures that have a herding tradition, which traces its origin all the way back to when we first started to domesticate animals for our own purposes and realized that we could in turn tap into their milk production from pregnant female mammals like cows, obviously, sheep, goats, camels, yaks, water buffaloes. The list goes on, everything other than humans, basically, that is a domesticated animal. We have somehow figured out a way to utilize their milk, depending on which culture is involved in the herding tradition and the milking tradition. And so in those cultures, a mutation has occurred close to where the gene for the lactase enzyme is found on chromosome number 2. And that has led to a continuance of the production of the lactase enzyme. Instead of the gene being disabled as you wean off milk and start consuming adult food, that gene continues to function because of this mutation. And it's a single change. And we know exactly what the change is in most individuals. And it has meant that instead of losing the ability to make the enzyme lactase and therefore to continue to metabolize milk sugar, lactose, you actually retain the ability to produce the enzyme and therefore to digest lactose. Now, as I said, a lot of people have lactose intolerance and don't even realize that they've got it because they have a mild form. But some of you have the not so pleasant form where if you even consume a small amount of dairy containing food, you have all sorts of digestive issues that follow on from that. And that's because while you may not be able to digest the lactose, the bacteria in your small intestine and down on into the colon most certainly can. And a byproduct of them metabolizing the lactose is that you have digestive issues. You have discomfort, gas, all sorts of things go on. and it affects your fecal matter. All sorts of things happen because you took in some milk-based products, and the bacteria in your colon are able to metabolize it, but the way they metabolize it is what leads to the discomfort. And towards the end of the semester, when we get into chapter 11, we'll talk about how the production of the enzymes for metabolizing lactose are actually produced in bacteria. We'll talk about how that's regulated, because it's an interesting system that allows the bacteria to metabolize lactose when it's present and to switch the system off when there's no lactose present, so they don't waste time producing enzymes for something that is not present, for a process that's not necessary at the time. So we'll come back and we'll readdress that system in bacteria when we get to chapter 11. But we're also going to come back and rethink lactose intolerance, or lactase persistence, to give it its proper name, again at the end of the chapter. But it is all about lactose and all about the fact that lactose is a member of one of the 4 classes of macromolecules. It belongs to the carbohydrate group that we're going to be talking about at length in this chapter. So that's why we have the lead in about lactose and about lactose intolerance, or as we ought to more properly call it, lactase persistence. So let me put this in here. Just put a So remember, the lactose intolerance affects about 70% on average. And lactase persistent So again, what we have to remember is that while using the term lactose intolerance implies that the loss of ability to metabolize lactose is somehow the odd thing, we need to really remember that as mammals, that's what really should happen. We should lose the ability to break down lactose and to digest lactose. So the unusual condition, as far as mammals is concerned, is lactase persistence. So that's why there is this tendency to focus on the persistence of the lactase enzyme rather than to put the focus on the people who lose the lactase enzyme, because they're really following the developmental pathway that mammals traditionally ought to follow, losing the ability to process milk sugar, because milk is, for want of a better term, baby food, and is not intended for adult mammal consumption. And we do see that reflected in the way humans process milk, because we don't continue to utilize our own human milk. We use the milk of other animals. And as I said, we milk a lot of animals. Cows, sheep, and goats are the common ones, but there are, horses, camels, yaks, water buffaloes, those are the major odd ones that you might not be aware of, that spring to mind immediately. I'm sure there are others that I can't remember right now, But we go to great lengths to get milk in order to continue to consume milk. We just don't consume our own past weaning. Not entirely sure why, but that's the way we've chosen to do things as we've developed as agriculturalists. So let's move on from here and start talking about the role of carbon in these macromolecules, now that we've raised this question of processing lactose. So once we get past the fact that most cells are made of water, about 70%, we need to recognize that the rest of the molecules are built on carbon skeletons. And these are referred to as organic substances or organic molecules. And the reason why is because up until the early 19th century, it was firmly held that organic molecules could only be produced by organisms, that humans could not artificially create organic molecules, that there was no way of doing this. And so the study of the molecules, these carbon-containing molecules from living organisms, became known as organic chemistry because it's the chemistry of life. And organic molecules were considered to be only formed by living organisms. And so if you had organic molecules present somewhere, that meant there were organisms around. And there was no way of separating organic molecules from organisms. And then in 1828, a German chemist by the name of Friedrich Wöhler accidentally made a substance called urea from non-organic molecules. Now, why this is a big deal is because urea is a major component of urine and is therefore an organic molecule. And it's got carbon in its skeleton, it's also got nitrogen and hydrogen, but it is considered to be an organic molecule and a product of life. And up until that point, Wöhler had been investigating urea and other components of urine as part of his research. And when he came into the lab one day and discovered urea crystals in a beaker that he knew he'd never had urine in, that was a big deal because suddenly he realized that he had made an organic molecule in the absence of life, in the absence of organisms. And knowing what molecules he'd had in that beaker, he repeated the experiment. And sure enough, now he doing it deliberately, he was able to confirm that this was in fact the way the urea was made. And he played with it a little bit and came up with a more efficient way of making urea. And we refer to this as the Wöhler synthesis. And it is still used today to make urea because urea is a component of synthetic fertilizers or artificial fertilizers. And it's a useful substance because it makes nitrogen particularly available to plants. They can take the urea up through their roots and use it for making all sorts of organic molecules that plants can make out of nitrogen. And that's how nitrogen gets into life, by being taken up by plants from nitrogen-containing substances like urea in the soil and being converted to proteins and nucleotides for nucleic acids, all of which we're going to learn about today. And then other animals, other organisms eat the plants to get access to the amino acids and other substances that contain nitrogen. It's how nitrogen gets into all organisms, by being taken up through the roots of plants from the soil. So it is being able to make urea was a big deal as far as Wöhler was concerned. It was a big deal for everybody because it meant that we were no longer limited to just the organic molecules that could be produced by life. We could go out and not only make the same molecules that organisms can make, and we do in abundance, but we can now make our own unique organic molecules. And this is the basis of our industrial chemistry. So all of the drugs that we utilize, all the synthetic compounds that we utilize, the gasoline that we burn in our car, and the other molecules that are derived from crude oil, these are all substances that are organic. They're built on carbon skeletons. And we would not be able to do all of this without Wöhler's accidental discovery of how to make urea, because no one would have considered that it was possible to make organic molecules synthetically, artificially, without life forms being present. And of course, that starts another discussion, because in 1859, Charles Darwin publishes On the Origin of Species, and Darwin and Wallace start the discussion on descent with modification, what we would call evolution today, and descent and natural selection. And of course, when you go back into the history of life, and we go backwards in time, back to what we refer to as deep time, at some point, we must come to the point where we see the very first cells to emerge as the origin of life. But then if we go one step further backwards in time, we must come to a point where there are no cells, just a soup of organic molecules. And the question that gets asked at this point is, could the chemistry of the early Earth prior to life appearing be capable of creating the molecules necessary for the formation of cells? And this was first proposed as a hypothesis in 1921 by a Russian biologist named Oprin and an English biologist named Haldane independently, and it's become known as the Oprin-Haldane hypothesis. And it was put to the test first in 1956 by an American graduate student named Stanley Miller, working at the University of Chicago. And Miller was able to demonstrate that using the model of the early Earth, first proposed by Oprin and Haldane, that it was in fact possible to synthesize simple amino acids that are found in proteins, simple sugars that are found in carbohydrates, and even some of the nitrogenous bases that we know are found in the nucleotide components of DNA and RNA. Not just with his first experiments, but with the experiments that Miller would continue to perform throughout the course of his life. He passed away about 10 years ago. And for the whole of his research career, he was a big proponent of the idea that the Earth's early chemistry was capable of creating these organic molecules that would lead to cellular life. There are other ideas that propose different origins of the organic molecules, or different locations on the Earth as to where the early chemistry might have taken place. But all of them propose that the formation of these organic molecules is perfectly legitimate in the absence of cellular life, that chemical conditions exist or existed here on Earth and likely exist elsewhere, not only in our solar system, but elsewhere in the galaxy, elsewhere in the universe, for the formation of these molecules, because we have found amino acids and other molecules that are related to cellular life in space, which tells us that even in what we might consider to be the vacuum of space, it's possible for the chemistry to take place. So the synthesis of organic molecules is not just a function of life anymore. It can be carried out independently of life so long as the chemical conditions are correct. And as far as we are concerned here on Earth, all of our industrial chemistry, or pretty much all of our industrial chemistry, hinges on our ability to be able to create organic molecules artificially or synthetically. So if that is the case, if we are in fact carbon-based, which we are, and these molecules are built on carbon skeletons, which they are, why is it carbon and not something else, given the chemical conditions that exist here on Earth? And the answer is two reasons. Number one, carbon can form 4 covalent bonds, and it can form 4 covalent bonds not only with itself, but also with the other 3 major elements that are found in life forms. And here you would have to cast your mind back to chapter 2 and remember that 96% of all the atoms in an organism can be accounted for by just 4 elements, carbon being 1, but the other 3 being oxygen, hydrogen, and nitrogen. And it turns out that all 4 of these elements can form covalent bonds with each other, and particularly with carbon. And so this ability to be able to form these 4 covalent bonds, not only with carbon itself, but with these other atoms, these other elements, means that it's possible to start building essentially an infinite number of different types of molecules. And that is part of the secret for why it's carbon and not something else. The other reason, if we accept that it is the carbon's ability to form 4 covalent bonds, which it is that when we look at other elements that ought to be able to do the same thing. So if you remember again back to our discussion for chapter 2, when we look at the periodic table, any elements that are in the same column have the same number of electrons in their valence shell and therefore have the same chemical properties. So the carbon sits at the top of column number 4, hence 4 electrons in the valence shell and the ability to form 4 covalent bonds. But there are other elements, excuse me, that sit in the same column below carbon. And they should also be able to form 4 covalent bonds, just like carbon can. So why is it carbon and not any of these other elements that form the basis for life as we know it? And the answer is because of the oxygen in our atmosphere. It turns out that while a covalent bond between 2 carbons can be attacked by oxygen, because oxygen is very reactive, and it can attack those covalent bonds, just take a little bit of cooking oil and put it in a glass and let it sit on a kitchen bench for a few days, and you'll see the changes happening as the oxygen starts to interact with the covalent bonds in the fats in the cooking oil, and it starts to change color and it takes on a funky smell as it goes rancid. But that's really the oxygen attacking the covalent bonds in the carbon chains of the fats in the cooking oil. So they're not perfectly stable in the presence of oxygen. But when these organic molecules are in organisms, there are protective measures in place to prevent oxygen from damaging the organic molecules in an organism. And in fact, life has taken it one step further over time and figured out how to take advantage of oxygen's chemical reactivity and use it to its own advantage in a process called cellular respiration, which we'll be talking about in a few chapters, where we take advantage of oxygen's reactivity to be able to generate boatloads of energy from the food molecules, the organic molecules that we consume. So life has not only figured out how to, number one, deal with the reactivity of oxygen and neutralize it, but turn it on its head and take advantage of that very reactivity to be able to make energy. But when we start looking at the covalent bonds between the next element in the periodic table below carbon, which is silicon, it turns out that the greatest reserve of silicon here on Earth is something called silicon dioxide, a silicon atom with 2 oxygens. And that you would know as dirt or sand, common garden variety sand. That's silicon dioxide. And that's because the covalent bond between 2 silicons is completely unstable in the presence of oxygen, and it immediately breaks down to silicon dioxide in the presence of oxygen. Can silicon form chains? Yes, but not in the presence of oxygen. It has to be in a non-oxygen atmosphere. So maybe somewhere else in the universe, there could be silicon-based life forms. Hey, there might have even been silicon-based life forms here on Earth early on, because oxygen is actually a product of life. So early in the Earth's history, there wasn't a lot of free oxygen around. In fact, there was virtually none. It was only when what we refer to today as oxygenic photosynthesis that produces oxygen, again, something that we'll talk about in an upcoming chapter, that oxygen starts to appear as a component first of the environment and ultimately part of the atmosphere. But until that happened, there was virtually no oxygen. So maybe early on in the history of our planet, there might have been an attempt to form a silicon-based life form. But any evidence of that disappeared once oxygen became a component because it couldn't survive in that environment. So there are two basic reasons why it's carbon and nothing else. Carbon's the sheer flexibility of carbon to form a huge array of organic molecules, and the relative stability of a carbon-carbon covalent bond in the presence of oxygen, the atmosphere that life exists under here on Earth. Could there be the possibility of weird life that is nothing like we know now? Absolutely. I've mentioned this before, I'll mention it again. Albert Einstein, one of my science heroes, made the statement about the universe that not only is it weird, and when we look around, we see that weirdness, but it's weirder than we can even imagine. And I suspect that applies just as equally to life, whatever other forms of life look like, other than our own. We know what life looks like on our planet, but that's because of the chemistry of our planet. But what if there is different types of chemistry that favor life, but not life as we know it? How would we even know that it exists? Because right now, we can only detect the life that we know using the tools that we have. I heard a piece by a biologist involved in the search for extraterrestrial intelligence the other day, and she was saying that there might even be the possibility, I guess people are thinking about this, that there is non-carbon-based life here on our planet, but we probably wouldn't know because we wouldn't even know how to look for it. It's an interesting thought. I hadn't even given some consideration to that, but that's even an interesting thought, that maybe there are different types of life here other than our own. We just don't realize it because we don't even know how to look for it, because we only know how to look for life like ours, like our form. Anyway, we're getting a little bit philosophical here, so maybe we should move on. So this diagram gives you some ideas about the flexibility of carbon, so we can form what are referred to as linear chains, which can be of many different lengths. In fact, we don't know how long a linear chain can get. It may be infinitely long. We can form branch chains, so rather than just being a long straight chain, you can put branches in. And the branches can be of many different forms. You don't have to link the carbons together by single covalent bonds. You could put in a double covalent bond. You could even put in a triple covalent bond between two carbons, and that would still be legitimate. It changes the nature of the molecule, and it changes the chemical properties. You can form circular molecules. This is a molecule called benzene. And you can see that it's got multiple double bonds in the ring. This is actually such an unusual molecule that there's a whole branch of organic chemistry that focuses on benzene and other things that are derived from benzene. It's a really key starting point for a lot of really interesting industrial chemistry processes. So this is an interesting molecule in its own right. Actually, let me give you some names of molecules here. So this is a molecule called ethane. This would be a molecule called 1-butene, and it's called butene for two reasons. Number one, it has the double bond in there, and that's where the ENE comes from. And then the but tells us that it's got 4 carbons in it. So this is just the way we name organic molecules. The F part here tells us that there are 2 carbons. The ANE tells me that they're linked together by a single covalent bond. So the way the molecules are named tells you something about it. This one is called 2-methyl propane. And the propane is because the longest continuous chain here is 3 carbons long. And then the 2 tells me that there's something attached to the number 2 carbon. That's the middle carbon here. And then the methyl tells me what's attached there. So this is a methyl group because that looks a little bit like methane. So if you can't remember what methane looks like, because I showed this to you when we looked at chapter 2, this is methane again. You can see that this looks a little bit like methane, but it's got one of the hydrogens missing, so we can form the covalent bond to the branch. So this is 2-methyl propane. And in fact, here's a model of it. We're going to talk a little bit more about this one in a little while. We'll talk about this one and a related molecule. And then, of course, I call this one benzene. So that's the name of this, this molecule. So you can see that there is a variety of different forms. So these are just basic variations on a theme. How can we link carbons together in different ways? And so you can see that these are some simple, but obviously various ways of linking carbons together. And we've not really even started to scratch the surface of the interesting ways carbons can be linked together to form molecules. But it illustrates the fact that carbon is extremely flexible in the types of basic structures, foundational structures that it can form. So this is our beginning of understanding where the variety of organic molecules comes from. It also helps us understand why there is such a great diversity of life, because there is this diversity of organic molecules that can be formed from carbon and nitrogen, oxygen, and hydrogen. So the diversity of organic molecules helps us understand the diversity of life. So we've talked, I just showed you methane, so we can move on from there. So here's methane again, if you want to see it, there's methane. You can see the different ways of drawing it. So my model is actually the ball and stick model, but it's a tangible model, and it's just something we've drawn on a page. This is the basic diagram that we would draw if we're drawing on paper. Remember, the dashes represent shared pairs of electrons, covalent bonds, and these are non-polar covalent bonds. So organic molecules tend to be non-polar, and that's part of a problem that we will get and we will have to discuss as we talk about carbon chemistry. And then you've seen the space filling model before, where the model actually illustrates the spaces occupied by the atoms that make up the molecule. So this would be the space filling model for methane. The only difference between methane and other organic molecules is the number of carbons in the chain. So in methane, there's a single carbon. When we start linking carbons together, then we start building larger and larger chains. And then when we start thinking in terms of molecules that we might be familiar with, one of the key molecules that's found in gasoline is something called octane. The oct tells us that there are 8 carbons in the chain, and the ANE tells me, as in ethane, that these are linked together by single covalent bonds, and then everything else is hydrogen bonded to that chain. And this would be referred to as a hydrocarbon, because it's a carbon chain with hydrogens on it. And because there are only single covalent bonds linking these carbons together, and then everything else is hydrogen, we would say that this carbon chain is saturated with hydrogen. So when we take a look at a typical fat, and particularly a fat that we might find in animals like ourselves, we find the same thing. Long chains of carbons linked together by single covalent bonds with lots of hydrogens attached to that chain. And each of these is called a fatty acid. They make up molecules that we actually refer to as fats. And we'll see how these are formed when we talk about the large group of macromolecules that the fats belong to, called lipids. We'll talk more in detail about this, but these fatty acids are saturated fats. Again, because they have a saturated carbon chain, just like the octane molecule. And if you burn octane, you produce carbon dioxide and water if you burn it completely, and you get energy out of it as you burn it. Hence the use of octane as a component of gasoline to provide energy for your car. Burning the gasoline, burning the octane releases energy that can then be used to drive the car down the road eventually. Same is true for burning fat and other food molecules. Again, they're organic molecules. They're largely hydrocarbons. And when we burn them, we produce a lot of carbon dioxide and water almost exclusively. And we release energy. But we don't burn it in the same way as we burn gasoline in an engine. Here it is a real fire, and it's actually an explosion. Here we have to be a little more subtle about the burning, because if we set our food on fire, we can't use the heat energy that's generated by literally burning something. So we have to break it down in the presence of oxygen, so it looks like a burning reaction, but we have to burn it, but we have to break it down in a more subtle way that releases the energy in a form that our bodies and cells in general can handle. And that's the subject of our look at cellular respiration in a few chapters' time. So more to come on the breakdown of these molecules, but these molecules must be broken down in order to release the energy that they contain. So similar processes, but not identical processes, allow us to access the molecule in these hydrocarbons. The properties of the hydrocarbon skeletons are altered then by attaching things to the carbon chains. And as we work our way through the macromolecules, we're going to look at the sorts of things that you can attach to the basic carbon chains in each case to change the chemical properties and create a unique set of organic molecules, the 4 different types of macromolecules that we're going to have to deal with as we look at the molecules of cells. So we start with this basic carbon skeleton with hydrogens on it, a hydrocarbon. It's non-polar. The problem with that is it doesn't dissolve in water. And for cellular chemistry, things have to be dissolved in water first. And so the addition of these additional groups that we attach to the carbon skeleton, which we refer to as functional groups, and you can see the name here, changes the chemical properties and by and large turns them from something that is mostly insoluble, and you may recall that the word for that is hydrophobic, water-fearing, insoluble, into something that is hydrophilic, water-loving, and therefore soluble and will dissolve in water. And so the functional groups and the understanding of the functional groups is somewhat important to our understanding of the broad diversity of organic molecules that make up cells. We don't dig very deep into the functional groups, but those of you who are going to go on and take some more advanced biology classes, which I know some of you are intending to do, you will learn more about the functional groups and their chemical properties and how they affect the large biological molecules in more detail in those more advanced classes. So, time for our first break. We've been going for a little while now. So here is our first concept check question. What properties of carbon explain the huge diversity of molecules organic or organisms can make? So if you want to go grab a drink, take a bathroom break, or you just want to take a little bit of time to think about what we talked about in the first section, by all means, put it on pause. I'll still be here when you come back. Otherwise, stick around for about 30 seconds and then I'll give you the answer to this question. Think through the question, see if you can come up with your own answer. Okay, welcome back. If you took a break, welcome back. If you just stayed around to wait for the answer, still welcome back. So what are the properties of carbon that explain the huge diversity of molecules organisms can make? Remember, there are 2 basic ideas that we need to address here. And I'm just going to move the text box over to the side so I have a little bit more room to write. So number 1 is, let's bring up the pen again. So number one, carbon can form four covalent bonds with itself and oxygen, hydrogen, and nitrogen. So that's number one. And number two, it can form a variety of skeletons. Linear, branched, circular, include double bonds. So it's this variety of skeletal structures that also add to the diversity. So it's these two things together that account for the diversity of organic molecules. Number one is that it can form four covalent bonds with itself and with, most importantly, oxygen, nitrogen, and hydrogen. And then the variety of forms of carbon skeletons that it can form is essentially infinite. But we know that there are linear forms, branched forms, circular forms, double bonds can be included, not just single bonds. We can have even a triple bond if we want. And the variety just goes on and on and on because of all of this. And so that's the reason why carbon, those are the two properties of carbon that account for the diversity of organic molecules that organisms are capable of making. So let's move on from there and start addressing the macromolecules, the really big molecules. So once we start thinking in terms of these organic molecules that life makes, we have to recognize that there are essentially really big molecules, macromolecules, that are the major components, the major carbon-containing components of cells. Three of the four classes of macromolecules are described as polymers. So these are all polymers. And we will address what a polymer is in upcoming slides, but I'm going to put this in here right now. These are all polymers, large molecules formed by linking together many small molecules. So carbohydrates, proteins, and nucleic acids are all recognized as being polymers. And what makes the difference between each of them is the type of small molecule that's linked together to create the really big molecules. And we call those small molecules monomers, and we'll readdress that shortly. There is a fourth class of macromolecules that's not mentioned here right now, but I'm going to mention it anyway. The fourth class of macromolecules are the lipids, or as some people refer to them, fats, which is technically correct. These are large, but are formed from molecules that were already large. And I'll add in there and not in chains, because one of the key features in polymers is that the little molecules, the monomers, are linked together in long chains. And that's not the case with the lipids, as you'll see. They're not they're formed from molecules that are already quite large, and they're not daisy chained together in long chains, as you would find in the polymer classes. So these are the 4 classes of macromolecules that we need to discuss in this section. So it says macromolecules of polymers, as I've just indicated on the previous slide. This is not necessarily correct. We can say 3 classes of macromolecules of polymers, and they're made by stringing together these small molecules called monomers. So this is just repetition. What's important here is to realize that the chemical reactions that assemble and disassemble the polymer classes of macromolecules are the same chemical reactions that assemble and disassemble the lipids, the fats. It's the same set of chemical reactions. So there is a reason why we include a discussion of fats in this section on polymers and macromolecules, because yes, fats are macromolecules. They're big molecules. They may not be polymers, but they're big molecules. And the chemical reactions that assemble and disassemble them are the same chemical reactions that assemble and disassemble the polymers. So it does make sense to consider them together. The 2 types of reactions involved are dehydration reactions. This is the assembly reaction. And hydrolysis, which disassembles. And they are essentially the opposite of each other. So dehydration, when you think dehydration, you think removing water, and that's in essence what happens in a dehydration reaction. So on this diagram, you can see the example of a dehydration reaction. So to make water, you need to take a hydroxyl group from one of the 2 molecules involved. In this case, it's attached to the polymer. That's not always the case. And you need to take a hydrogen off the other molecule that's involved. In this case, it's on the monomer, but again, that may not necessarily be the case. It depends on the polymer and the monomers that are involved as to which comes off which of the 2 molecules. But in this example, it's the hydroxyl group coming off the polymer and the hydrogen coming off the monomer. So those 2 together give you the water. So these 2 together are what are giving us the water. And what we are doing is breaking the existing covalent bonds between the hydroxyl group and the polymer and the hydrogen group and the monomer, and creating a new covalent bond that links the monomer to the polymer. Let's clean that up a little bit, I think. So that's what the dehydration reaction does. Hydrolysis reverses that. It breaks the covalent bond that links the monomer to the polymer chain, and in the process adds back the functional equivalent of water. So we add back a hydroxyl group to one end of the broken covalent bond, and we add the hydrogen back to the other broken end of the covalent bond. So the term hydrolysis, hydro means water, lysis means to break, so it literally means to break with water or to split. We'll put in here that it breaks the existing covalent bond. And I can give you a really good example of this one. This is digestion. If I give you any, let me put an example in here. It won't mean a whole lot right now, I suspect, because we haven't talked about making proteins, but we will talk about making proteins just a little bit in this presentation, but later in the semester when we talk about two processes called transcription and translation. Translation is actually all about making proteins, and we'll talk in more detail about how that's done then, but this is establishing our foundation for that. So this animation you will find in the on the resources page. All the animations that are included in the PowerPoint are on the resource page, and this is the animation on dehydration and hydrolysis reactions. So in the interest of time, and because you can play these animations separately from this presentation, I'm not going to actually run the animations within the presentation because that's only going to stretch out the presentation. It's going to be reasonably long as it is without running all the animations. And you can sit down and watch all of these animations in your own time to get a better feel for each of the processes that are described here. So this animation walks you through the formation of polymers via a dehydration reaction and the disassembly of polymers via a hydrolysis reaction. So an animation that you might want to look at. So in terms of getting access to the monomers that we need to make our own macromolecules, we really do need to break down the existing macromolecules that we take in our food. And that's why digestion is a really good example of the breakdown of macromolecules and a really good example of hydrolysis, because all of the molecules that you take in that we call food molecules, so lipids, fats, proteins, and carbohydrates are all macromolecules that need to be disassembled into their respective components, monomers if they're polymers, the other components when we're thinking about the lipids, so that we can assemble our own forms of macromolecules, our own proteins, our own carbohydrates, our own fats, because we can't make these, the smaller component molecules very easily, or can't make all of them. We may be able to make some of them, but we can't necessarily make all of them. We have to get them in from our food, and we have to liberate them by breaking down the large macromolecules of our food in order to be able to get access to those smaller molecules for assembling our own macromolecules. That's the second reason why we eat. The first reason why we eat, to get energy. So we're going to be addressing these two ideas as ongoing components of the class in some upcoming chapters as well. So, quick break. I know we haven't covered much ground as far as the polymers and monomers are concerned, but I think it's important to stop at this point just to remind ourselves again via a concept check question what it is that we're talking about. So most of the very large molecules in cells are called something because they are assembled from smaller molecules called something. The chemical reactions that assemble these molecules are called some type of reaction, and the reverse reactions are called some other type of reaction. See if you can come up with the terms to fill in the blank or blanks. So most of the very large molecules in cells are called polymers. Because they are assembled from smaller molecules called monomers. Chemical reactions that assemble them are called dehydration reactions. And the reverse reactions are called hydrolysis reactions. So if those were the terms that you came up with, good. You have an understanding of that little section of the presentation. So onwards and upwards. So here are the four categories again of the large biological molecules, otherwise known as macromolecules. Remember again, carbohydrates, proteins, nucleic acids. These are all polymers. The lipids, of course, not polymers. So as we talk about the different types of macromolecules, we'll talk about, in the case of the polymers, what the monomers are. And we'll look at the different types of monomers that create the different types of polymers. And of course, we'll address the molecules that are assembled together to form lipids as well. First group we're going to talk about, carbohydrates. Now, carbohydrates have, over the course of the last few years, been given a bad rap. And this happens periodically. The idea of a low-carb diet, so the Atkins diet, South Beach diet, any of these diets that emphasizes reducing carbohydrates in your diet, in your food intake, these are not new. They've been around for 150 years or so under different names. And they keep coming back periodically. About once every 30 or 40 years, someone, quote unquote, rediscovers a low carb diet or a low fat diet or a low protein diet. There are variations on different types of diets. The problem with all of these diets is they emphasize cutting out one of the major macronutrients, the fats, the carbohydrates, and the proteins. The reality is we do require all of them. We do require carbohydrates. We do require proteins. We do require lipids because they provide us with the raw materials for creating our own macromolecules that are carbohydrates, proteins, and lipids. And the reality is that if we don't take in carbohydrate, our body will preferentially try and create carbohydrate from some of the raw materials that we take in the other forms. It doesn't do it very effectively, it doesn't do it very efficiently, and it is not its preferred way of getting carbohydrate. And the reason why it requires carbohydrate is because this is what cells prefer to metabolize as a source of energy. And therein lies one of the problems with the carbohydrates. The carbohydrates that are problematic are what are referred to as simple sugars. Small sugars that are easy to metabolize and yield only calories and nothing else. And because we tend to consume way too much of those, we tend to take in a lot of calories without getting much other benefit from them. In fact, getting no benefit from them other than calories. And the problem is that our bodies are tuned in such a way that any excess calories that we take in are not discarded. They are stored, and they are stored in the most energy dense form of macronutrient that we have, fats. Fats are twice as energy dense as proteins and carbohydrates. So because animals need to be mobile and move around, it makes sense to store the excess calories in something that allows you to store a lot for very little weight or very little mass, and that's fat. So if you consume a lot of calories in the form of sugars, particularly simple sugars, that provide you with nothing other than lots of calories, but no other nutritional benefit, then not surprisingly, you are going to lay on a lot of fat because your body is simply not going to discard those excess calories. And that's why carbohydrates need to be treated carefully. It is inaccurate to say that you need to limit the amount of carbohydrates in your diet. That would be bad news because your body needs it. And if you've ever tried to limit carbohydrates, you know that sooner or later you give in and you pig out on carbohydrates because your body is basically saying, I need carbohydrates, and you're limiting those carbohydrates to me is just not on. I need those carbohydrates. And that is the problem also with these fad diets that focus on excluding one of the macronutrients and focus on maximizing the other ones. All of those diets are not diets that you can maintain for life, because the original use of the term diet was really to talk about what you are going to consume on a day-to-day, week-by-week, year-by-year fashion for the whole of your life, not just for some short period of time while you moderate something or attempt to lose some weight. Because weight management is not something that you do for a little while, it is something that you do for a lifetime. And for some of us, it is more of a struggle over a lifetime than it is for others. But we will all have to practice weight management at some point in our lives, because that is the way our bodies operate, unfortunately. So saying that we want to exclude carbohydrates or we want to limit the amount of carbohydrate is to focus incorrectly on how we should treat carbohydrates. What we should be doing is limiting the amount of simple carbohydrate and focusing more on consuming complex carbohydrate that can be partially digested, that can provide an energy source for cells in the form of glucose, blood sugar, but is not going to provide us with just a source of energy, but he's going to provide us with other forms of nutrients too, because of the way in which we take it in. So let's talk a little bit more about the nature of carbohydrates. So the monomers, the building blocks for building the complex carbohydrates, are what are referred to as monosaccharides. So you understand the prefix mono, it means one. Saccharide comes from the Greek word for sugar. So a monosaccharide is simply a single sugar. And these are the smallest of the sugars. These are the ones that are typically used for energy. So glucose is what we refer to as blood sugar. And it is the primary fuel that all of our cells and our body in general prefers to utilize as a source of energy, particularly the brain. So what may surprise you is that while your brain constitutes a small part of the mass of the body, less than 10%, it actually consumes well over 30% of all of the energy that the body consumes. And its preferred fuel is glucose. It is one of the molecules that can cross what is referred to as the blood-brain barrier. Not all molecules that circulate in the bloodstream can cross into the brain. Glucose happens to be one of them, and it serves as the primary fuel that our brain cells, the nerve cells that make up the brain, prefer to use. So these sugars, glucose and fructose, are going to be sugars that we are going to encounter again when we start thinking about cellular respiration, because it turns out that glucose is converted to fructose, which is then broken down to smaller sugars as part of the starting part of cell respiration. So we are going to see glucose and fructose involved in cell respiration. We're also going to see them involved in the more complicated sugars, particularly glucose, but to some degree fructose as well, in forming some of the more complex sugars that we need to pay attention to. Now, one of the issues that we have in considering sugars in general is that we eat a huge amount of sugar on a yearly basis, on average. Our textbook says the average person consumes about 100 pounds of sugar in America every year. I've seen numbers as high as 140 pounds. It used to be over 150 pounds per year. So we're doing a little bit better in terms of our average sugar consumption. But if you were to look at that in terms of bags of sugar that you would find that you would buy from the supermarket, the old sugar bag used to be a 5-pound bag. It's now a 4-pound bag. But the old 5-pound bags, for 100 pounds of sugar, you would need 20 bags of sugar to make up that 100 pounds of sugar. And no one in their right mind would eat that much sugar at one sitting. So how do we manage to consume that 100 pounds of sugar? Well, it's because sugar is found in all sorts of food that is processed. Most processed food contains one of three things, usually all three of them, but it contains salt, sugar, and added fat. Added salt, added sugar, and added fat as part of the processing. That's why you pay for it. It's what's called value added processing. And the value that's added is in the form of the sugar, the fat, and the salt that's added to the food as part of the processing. That's what you're paying for. So bear that in mind as you think about it. What sort of foods might you find sugar in that you're not expecting to find it in? Ever looked at peanut butter? You would think that peanut butter would have peanuts and maybe a little bit of salt, and that would be it. But no, peanut butter has sugar in many different forms. And that's one of the other problems. Typically, when we think of sugar, the names of most sugars end in OSE. So if you look at, well, we've got some examples here, glucose, fructose. If we think about table sugar, which is sucrose, when we think about the milk sugar, which we saw was called lactose, notice that they all in OSE. So you might find dextrose. It's just another fancy name for glucose. But if you see and -OL name, like mannitol. Another one, common one is sorbitol. These are sugar alcohols and are just as sweet. And just as problematic, they bring just as many calories with them as the sugars themselves. Then one of the other problems is that food manufacturers are coming up with all sorts of alternative names to try and disguise, I guess, how much sugar is actually in there. So if you see something like agave syrup, well, that's just a replacement for honey. And remember, honey contains both glucose and fructose, as it says there, and not much of anything else, by the way. It's a very concentrated sugar syrup, as is agave syrup. And they're just used as a way of adding sugar for sweetness. And it's still just sugar, and it's still just empty calories. Likewise with high fructose corn syrup. Why was that added? Because it was cheap, it was easy to make, and it was a lot cheaper to use than table sugar, sucrose. And so it could be added to all sorts of foods without costing a whole lot. And of course, we are sugar addicts. And so anything that's got sugar in it, we tend to want to eat. It is known that to get a child to eat a new food that's sweet requires just a single exposure, and that's usually enough. For something that is non-sweet, it might take up to 10 exposures to the food to get a child to willingly eat the food and to enjoy it. We are sugar addicts. Let's be honest, that's what we are. So another way of sneaking sugar in under the radar screen, if you ever see on the ingredients list something called dried cane syrup, you're dealing with sugar, with sucrose. It's just table sugar, because that's how we get the sugar in the first place. We squeeze the juice out of sugar cane. We allow the water to evaporate, and what crystallizes out is the raw sugar. We then spin the molasses out of it, and redissolve and recrystallize the remainder, and that's what gives us the nice white sugar crystals that we're familiar with from the supermarket. Ironically, most sugar producers are now selling brown sugar or turbinado sugar, and what they do is take the white sugar and add a little bit of molasses back to give it the brown color and the flavor, having already taken the molasses out in the first place. Seems kind of pointless to do that, but that's what they do to give us a product that we think is healthier, and therefore something that we would be more willing to buy. Is it healthier? Probably not, because molasses is still just pure sugar. Different form of the sugar, but it's still just pure sugar. Anyway, these are the monosaccharides. And these are the ones that we need to limit, because these are the ones that bring a lot of hollow calories and not much other nutritional benefit. So it mentions that glucose and fructose are isomers. So we need to stop for a moment and just discuss isomers in a little bit more detail. So here is a picture of glucose and fructose, and these are referred to as isomers because if you count up the number of carbons and the number of hydrogens and the number of oxygens, they are the same in each of them. And you can see the chemical formulas here. C6H12O6, 6 carbons, 12 hydrogens, 6 oxygens in both of them. But they are clearly arranged into different structures. So these are referred to as structural isomers. And the next animation, which is on the next slide, actually walks you through the 3 different classes of isomers that we recognize. Structural isomers, geometric isomers, that are also known as cis-trans. And enantiomers that are also known as mirror image isomers. So I've got my models here and I can show you examples of the isomers as well with my models, but I would strongly suggest you go and take a look at the animation. Remember, again, the animation can be found in the resource page for this chapter, which is chapter 3. So if you take a look at the resource page, you will find all of the animations listed in order as they appear in the PowerPoint presentation. So here are 2 structural isomers, and these are the ones that are actually pictured here on the screen. So this is the linear version of the molecule. So you can see this here, and I'll put the cursor on. This is the diagram of the linear version. So it has the formula C4H10. This is the branched version, which is this one. It is again C4H10, but clearly it's a different structure. And you might recognize this one because this is our old friend 2-methyl propane. This is called N-butane because it's got 4 carbons in it, and that would be a bute. And with the single covalent bonds, that would be butane. But with C4H10 being the shared chemical formula, these are clearly different structures. So these are structural isomers. The next, the geometric isomers, I have my models. So this one is this form, and this is called the trans form. So maybe I'd put a label on that. This is the trans form. And here's the other one. You can see that these 2 are very different. And this one is called the cis form. So trans means opposite and cis means same. So here, these are on the opposite side of the molecule. These are on the same side. So trans means opposite, cis means same side. And these are not equivalent. You can't just twist this around to get this. These are very separate, and the only way to convert between these is to carry out a chemical reaction. And these are going to become important when we talk about lipids and fatty acids a little bit later in this presentation. What gives the possibility of these? A double bond between the 2 carbons. So you can see that these carbons are linked together by a double bond, and that's what locks these 2 molecules rigidly in these forms and gives you the possibility of cis and trans isomers. So these are the cis and trans isomers. And again, the key feature, a double bond between the two carbons that are involved here. We'll see those play out a little bit later in the presentation. Then the last type of monomer is called an enantiomer, or a mirror image isomer. And so here are my two models, and I'm going to show you each one separately and hold them the same, and then show you them side by side. So here's number one. I'm holding it by the orange ball at the top. You can see that up there. Then you can see that there's a brown and a blue and a white ball also attached to the carbon in the center, because the black ball is the carbon in the center. So that's number one. Here's the other one, holding it again by the orange ball with the white ball at the back. And this time you can see that the position of the blue and the brown have been flipped from one side to the other. Here you can see them side by side. And what makes them mirror images, let me get rid of that again, somehow that turned up. What makes these mirror images is because if you just take them and slide them sideways like this, nothing really matches up. But if you take them like this and put them together and then rotate them like that, you can see that everything matches up with the partner on the other molecule. Now, why does this represent mirror image? Because if I take my hands, they behave exactly the same way. If I slide them over like this, nothing matches up. But if I put them thumb to thumb and then rotate them, all the fingers match up with their partner fingers on the other hand. And the reason why they're mirror images is because if I hold up my right hand to the camera, what I see in the camera is an image of my left hand. So if I hold up my left hand away from the camera and look at it, it looks just like the image of my right hand that I see in the camera, because they're mirror images of each other. That's why these molecules are considered to be mirror images, or mirror image isomers. And what makes the difference between the two is the fact that there are 4 different colored balls, an orange, a white, a brown, and a blue, or purple, depending on how you want to interpret the color, attached to the central carbon. That's what makes the difference. This carbon is described as being asymmetric. So this is the third class of isomer, the mirror image isomer. So those are the 3 classes of isomers. Structural isomers, and glucose and fructose are structural isomers. Geometric isomers, which we'll encounter when we talk about the fats. And enantiomers, or mirror image isomers, which we're going to see when we talk about proteins. But we could talk about enantiomers with some of the other molecules as well. But when we talk about proteins particularly, we'll be able to talk about enantiomers in a little bit more detail, because it's really obvious where that comes into play there. So take a look at this animation from the resource page so that you can familiarize yourself a little bit more with the nature of these isomers, because it's really important to have a basic understanding of these 3 types of isomers. And then this one discusses a molecule called dopamine, which is at the heart of understanding Parkinson's disease, because it turns out that dopamine also comes in 2 enantiomers. One is referred to as L-dopamine, or L-dopa, and the other is referred to as R-dopamine, or R-dopa. It is L-dopamine that is absent in Parkinson's disease. The cells that make dopamine in the form of L-dopamine in the brain stop making it, and that leads to Parkinson's disease. And it is only the L-dopa form that is produced by those cells and therefore is biologically active. And because of the way the cells carry out their chemistry, they only make the L-dopamine. But we can synthetically make dopamine as well. But when we make it synthetically, we make both the L version and the R version. And we can use that dopamine to treat Parkinson's disease. It's not a perfect treatment, and over time, the dopamine becomes less effective and the full symptoms of Parkinson's disease return. But the reason why we can do this is because the R-dopamine has no effect. It's biologically inert, which is good news, because it's present with the L-dopamine because of the way we carry out the chemistry. Very, very different from the way cells do it. And so our industrial chemistry makes both mirror image isomers, the L version and the R version. The R version treats the Parkinson's disease. The R version is there, but fortunately, it is biologically inert. With other drugs, going back into the history of drugs, and particularly back into the 1950s and 1960s, that has not always been the case. And if you want to look up a really good example of this, do a Google search on the drug called thalidomide. So the transcript service is having trouble with the term thalidomide. So let me spell that out for you so you can look it up. So look up thalidomide. So this is a great example of where knowing something about the two isomers really mattered. So look up thalidomide. In the interest of time, I'm not going to talk about it, but if it's a very sad story, and I would strongly advise you to take a look at the thalidomide story. Okay, we need to move on with carbohydrates. That was a little bit of a diversion, but it's one of the diversions that the textbook takes, or detours that the textbook takes in talking about organic molecules. So again, onwards and upwards with carbohydrates. So monosaccharides, like glucose, primary energy fuel for cells. But we want to look at the polymers as well. So what we know about the monosaccharides is that not only do they come in the linear form that we've been drawing, they also come in a circular form. And the circular form is the key for understanding the polymers, because it's the circular forms that are linked together. You don't need to know how it takes place, just that it does, and that there are linear and circular forms of the sugars, particularly the 6-carbon sugars like glucose and fructose, which are the ones we primarily focus on. So once we understand that we have this ring form, let's take a look at how we link them together. So not surprisingly, it's going to be a dehydration reaction. The simplest forms are known as disaccharides, where we take 2 monosaccharides and link them together. So the milk sugar that we've been talking about, lactose, that's a disaccharide formed from the sugar glucose and another related sugar called galactose. Maltose, which is the cereal sugar, is 2 glucose molecules linked together. Sucrose, or table sugar, is glucose and fructose. So this is 1 glucose and 1 fructose. So that's what those disaccharides are made up of. So this animation walks you through the formation of a disaccharide, and specifically the disaccharide is sucrose. So there's the 1 molecule of glucose and 1 molecule of fructose that are going to form the sucrose disaccharide, and it'll show you how what is known as a glycosidic linkage is formed. A glycosidic linkage is the chemical bridge between the 2 sugar molecules as they are linked together to form the polymers. And in the case of sucrose, it's going to be what is known as a 1,2 glycosidic linkage. And that is one that pretty much every organism on the planet has the capacity to break and therefore can metabolize sucrose, table sugar, because sucrose, it turns out, is also the main way in which plants transport sugar around within a plant. And some plants, like sugarcane, produce so much excess sugar that they store it as sucrose as well, and we simply extract it out and use the sucrose, the table sugar, for our own purposes. But again, as I've previously pointed out, this animation is available on the resource page, so I'm not going to play it here in the interest of time, but you can take a look at this on via the resource page. There's a direct link to it so that you can run this. And again, they're in the order in which they appear in the PowerPoint. So take a look at this animation to see how glucose and fructose are linked together to form sucrose and the formation of a glycosidic linkage. So here's how we would draw a simple glycosidic linkage. So this is forming lactose, the milk sugar. Again, it's a dehydration reaction. So we remove a hydroxyl group and a hydrogen group. That gives us the water. This is a dehydration. And we're forming a glycosidic linkage. That's the oxygen bridge between the two sugar molecules. And again, the animation on the previous page will show you how this is formed with sucrose. We've talked a little bit about sucrose. We've talked a little bit about high fructose corn syrup. There are actually three forms of corn syrup. There is regular corn syrup, where the ratio of fructose and glucose is pretty much the same. It's as it's extracted out of the corn. There's a low fructose corn syrup that has more glucose than it has fructose, and you can create that by converting some of the fructose to glucose. They're interchangeable. And then there's high fructose corn syrup, where you change the glucose to fructose, or some of the glucose to fructose. And it's all about manipulating the ratio of glucose to fructose through chemical manipulation. And as I said, high fructose corn syrup was used as a sweetener and is still used as a sweetener because it's cheap, it's easy to make, it costs so much less than sucrose. Sucrose is relatively expensive compared to high fructose corn syrup. And so for food producers that wanted to add a sweetener, high fructose corn syrup was the preferred one because it was so easy to get. People have argued that high fructose corn syrup is somehow less healthy for you than table sugar. The reality is it's not. The ratio of glucose to fructose does not really matter. What really matters is the fact that you're getting so much sugar and not much of anything else. You're getting so many calories in the form of simple sugars in the corn syrup that you are going to get more calories than you need, and you're going to start laying down those extra calories as fat. It leads to people getting overweight as a consequence of consuming all of these excess calories in the sugars. The ratio of glucose to fructose does not really matter as much as the fact that you're getting so much sugar and so many calories in the form of sugar with virtually no benefit other than lots of calories. That's what the real issue is with high fructose corn syrup. Changing to sucrose, sucrose is really no more natural than corn syrup. They're both processed out of plant material as a source of sugar. It just sounds more natural because we consider sucrose to be somewhat less processed, but in fact, it goes through a lot of processing, almost as much processing as the corn does to get the high fructose corn syrup. So in terms of nutrition, there's very little difference. There's very little difference in terms of calorie content, way too many calories. we just have been tuned to the fact that high fructose corn syrup is more commercial than sucrose. Sucrose is somehow more natural, and I'll put that in air quotes. But in reality, they're all just lots of sugar with lots of calories and not much of anything else. So we talked a little bit about sugar consumption early on. I'm not going to rehash this. We've already covered that. So the last type of polymer are the polysaccharides. These are the really long chains of monosaccharides. There are three of interest to us. Starch and glycogen basically serve the same purpose in the organisms where you find them. So starch is found in plants and is a way of storing excess sugar and glycogen is a way of storing sugar for easy energy access. In muscles and the liver. This is where we store our glycogen, and glycogen is found in animals. So these are ways of storing energy. And the sugar, by the way, is glucose. So both are bulk polymers of glucose. Then the third polymer is cellulose. And cellulose is formed from a different form of glucose. There are two forms of glucose. And because of this, the cellulose fibers are hydrogen bonded. So cellulose is the primary ingredient in wood. It's the reason why wood is so strong. The other thing about cellulose, and that's mentioned up here, is that because of this slightly different form of glucose being used, the link between the glucoses is a little bit different. And it turns out that that link, unlike the link between the glucoses in starch and glycogen, which can be broken down by most organisms, the link between the glucoses in cellulose can't be broken down. So it can only be digested by some prokaryotes. and fungi. So here is a form of carbohydrate that is actually beneficial because being found in fruits and vegetables, it's indigestible, it provides volume so you feel full, but limits the number of calories that you take in because you can't break this carbohydrate down. And so if you are on something like the Atkins diet or the South Beach diet, you wouldn't be eating a lot of fruits and vegetables because there is a trend to get away from that. And you're only allowed to eat certain types of fruits and vegetables. And you're actually limiting the variety that you need to make your food interesting. And in fact, fruits and vegetables are a really good source of nutrition. Lots of animals eat them. All herbivores eat them. And they make a very good living out of it. And our digestive tract is actually designed to eat a combination of fruits and vegetables and animal products. We are what is described as an omnivore. That means we do eat plant material and we do eat animal material. And our digestive tract can handle both. And so taking in fruits and vegetables, getting the cellulose, getting the fiber in our diet, it is beneficial. It's known to inhibit the uptake of fat, to inhibit the uptake of cholesterol, to provide volume in our diet so that our digestive tract moves and the food in it moves through freely. It allows for absorption of a little or retention of a little bit of water to lubricate the movement of our waste through and make sure that everything actually works the way it ought to work. There's a lot to be said for taking in fruits and vegetables and taking in the cellulose that makes up the cell walls of the fruits and vegetables, the cells of the fruits and vegetables. There's a lot of value in eating plant material, and we should not discount the carbohydrates that are here, because the carbohydrates in fruits and vegetables, even the glucose and the fructose that are found in fruits and vegetables, are found in limited amounts compared to what you might get from processed foods. Again, eating an orange only gives you about 90 calories for an average orange, whereas drinking a glass of orange juice could provide you with nearly 300 calories, depending on the type of fruit juice that you're drinking. And that fruit juice has lost all the fiber because you've taken the pulp out of it. The pulp is the fiber. So you're actually losing possibly the best part of the orange juice by removing all of that pulp. Again, it's not the nutrition that you're getting, it's how you get that nutrition that matters. So eating the unprocessed fruits and vegetables is much better than eating the processed or drinking the processed fruit or the processed juice. Again, it's all about thinking through and what you're doing and following a simple rule: all things in moderation, nothing in extreme. So this shows you diagrams of starch, glycogen, and cellulose, and you can see the differences in the molecules, and yet they are all polymers of glucose. Starch and glycogen, very, very similar, using the same type of glucose molecule. In cellulose, it's the alternative form of glucose that allows for the hydrogen bonding. That is what changes the properties of cellulose compared to starch and glycogen. So that's the carbohydrates. So time for another break. We've been going for a while again. So when we think in terms of carbohydrates, just to double check that we remember what carbohydrates are all about. All carbohydrates consist of one or more, sometimes referred to as simple sugars. Table sugar or something is an example of a particular type of sugar. So let's stop for about 30 seconds. If you're going away for a bathroom break or a drink or a snack or something like that, put us on pause, come back and start up again when you get back. So all carbohydrates consist of one or more monosaccharide. Sometimes referred to as simple sugars, table sugar or sucrose is an example of a disaccharide. Okay, so onwards we go to the next type of macromolecule, the lipids. Lipids are almost pure hydrocarbon, and they are extremely hydrophobic. But that's kind of important because of the role that some of the lipids perform in cells. We want them to actually be hydrophobic, insoluble in water because of the role that they play. So when you start thinking in terms of fats, you think in terms of things like butter, margarine, olive oil, canola oil, cooking oils in general. You might think of Crisco. These are all examples of lipids or fats. When we start thinking in terms of the types of molecules that lipids are formed from, remember they're not polymers. They are not daisy chains of lots of small molecules. They are large molecules because they are formed from molecules that are themselves relatively large, but they're not daisy chained together, as you're about to discover. So when we think in terms of a fat found in human bodies, they are a specialized type of lipid. They are a lipid that contains fatty acids. So fatty acids are the subunits that are typically found in those molecules that we refer to as fats, or to give them another name, there are two other names. So they are sometimes known as acyl glycerols or glycerides. Those are the fats that are found in our body. At the heart of them is a skeleton of a skeleton formed by a molecule called glycerol, and to that is attached some large hydrocarbon molecules called fatty acids. And the most common form of fat in the human body and in the bloodstream, actually, I'll put it this way. Let's do it in black. So if you've ever had some blood tests done for your doctor, he may have talked or she may have talked to you about triglycerides. Those are the fats that circulate in the bloodstream, but they're also the fats that make up the vast majority of fat in our body. So those are the ones that we're going to focus on primarily here. So let's take a look at what they look like. So this animation will actually walk you through the assembly of a triacyl glycerol or a triglyceride. So here you can see the glycerol backbone that the fatty acids are going to be attached to. Here you can see some typical fatty acids. These fatty acids are referred to as saturated fatty acids. So let me put this in here. This is the reason why they're referred to as saturated, because they have the maximum hydrogen atoms attached to the carbon chain. So we actually say that the carbon chain is saturated with hydrogen. And what gives it away is the fact that you've got single covalent bonds between the carbons. That tells you that you've got a saturated carbon chain here. And you can see that as a consequence of this, they are linear in shape. And so as a consequence, they can pack really closely together. So saturated fats, of which this is an example, tend to be solid at room temperature. Let's put that in here. They're solid at room temperature and mostly come from animals. So if you think about the sorts of fats that you're aware of that are solid at room temperature, the first one that most people think of is butter. The second one would be pig fat, or what is known as lard. That's the white fat that's left over in the fry pan after you cook some bacon for breakfast or something like that. Beeswax, tallow, which is a beef wax, and suet, which is another beef wax. It comes from cattle. These are all solid waxes. Tallow and beeswax have been used for candles. Now, beeswax is still used for candles in many cases. So these are all examples of saturated fats. They're solid at room temperature. And that's classic of fats derived from animals. And so this will show you how a saturated fat is, or a saturated triacyl glycerol or triglyceride is formed. And the bond between a fatty acid and the glycerol is called an ester linkage. And as you might guess, it's formed by a dehydration reaction, as you'll see in the animation. So again, this animation is available through the resource page for this chapter in Canvas. So here you can see an example again of a fatty acid and the glycerol backbone. So here's the glycerol backbone. It is a dehydration reaction, so that's going to be removed. There's the water molecule coming out. Here is the example of an ester linkage. And again, This is a saturated fat. This is a saturated fat. This is an unsaturated fat. So how do I know it's unsaturated? The giveaway is the presence of the double bond here. So the double bond means unsaturated. But more than that, you can see what it does to the carbon chain. It puts a kink in it. And that kink means that it's going to occupy more space. And so these are not going to pack as tightly together, and they tend to be liquid at room temperature. So fats containing unsaturated fatty acids tend to be liquid at room temperature and come from plants. So all of those cooking oils like canola oil, olive oil, vegetable oil, safflower oil, olive oil, the list goes on. Yeah, I mentioned olive oil already. The list goes on and on and on. Those are all plant-derived fats, and they tend to be liquid at room temperature because plants make a percentage of their fats as unsaturated fats. And what I should mention here is, in nature, these cis isomers. So hopefully you'll remember from the previous section, we talked about cis trans isomers or geometric isomers. Unsaturated fats in nature, naturally occurring unsaturated fats, are of the cis form, and that's what gives them the kink. And there's a story in that we will get into very quickly. So animal fats, we've already talked about, tend to be solid at room temperature because they pack closely together. They're linear fats. Unsaturated fats tend to be found in plants, but also in, and I'm going to put an arrow here, particularly cold water, fatty fish, like salmon and tuna. And the reason why they have these unsaturated fats is because those unsaturated fats act as antifreeze and stop the blood of the fish from freezing in really cold water. And it turns out that those fats, when we consume them, are heart protective. They actually protect us against heart disease and stroke. They are very, very useful under those circumstances. And that's why we should eat fish. We're encouraged to eat cold water fatty fish 2 to 3 times a week in place of lots of other fatty animal-derived protein that has a lot of saturated fat in it. So if you're going to eat meat, make sure that you're eating lean meat that has low levels of saturated fat in it, or that you are eating cold water fatty fish that has the heart protective unsaturated fats present in the fat content of the fish. So here's some of the locations where you can find saturated fats. And here's where you can find some unsaturated fats. Now, the ones that we need to be careful of that are unsaturated fats but are dangerous to us, the trans fats. So remember again, we've got the cis and the trans form of geometric isomers. The naturally occurring unsaturated fats are of the cis form. But when food manufacturers first started to use plant-derived fats in food, they discovered that they wouldn't give the same texture as saturated fats like butter and so forth, or frying oil for French fries that was derived from beef fat and lard and pig fat and things like that, saturated fats. Because if a fat is already a liquid at room temperature, you can't heat it up to very high temperature before it starts to burn. We call it smoke temperature. A solid fat needs to be raised in temperature just to turn it to a liquid, and then it can be raised to an even higher temperature in order for frying, for deep frying. And so you need that for creating the perfect French fry. You also need saturated fats to create the perfect chocolate chip cookie that's soft and chewy and not the texture of a hockey puck. So the way the food manufacturers figured that they could get around this was to bubble hydrogen through it, a process that is called hydrogenation. And that converts some of the unsaturated fats to saturated fats and creates a plant fat that has a consistency a little bit closer to a saturated fat. It's not as liquid, it's a little bit more viscous, it's a little thicker, it can tolerate more heat, and it gives a French fry and a chocolate chip cookie that is closer to what you would expect to get if you used butter or animal fat for doing the food preparation. The problem is that when you partially hydrogenate or hydrogenate, so hydrogenation converts some cis fats to trans fats. And the effect is to go from a fat that has a kink in it like that to an unsaturated fat that goes like that. This is cis, this is trans. And so this behaves more like a saturated fat. Because trans fats are almost always unnatural, they're the product of human processing. There are a few examples of trans fats in nature, but they're very, very limited and you would not encounter them. So if there's trans fat in food, it's because humans have created it as part of the processing, typically by hydrogenation of unsaturated plant fats. And that poses a problem because being unnatural, we don't know what a healthy level of trans fats looks like. And in fact, the evidence that we have right now suggests that trans fats are just as unhealthy as saturated fats. In other words, they promote heart disease and stroke. And in fact, we think they may be more dangerous than saturated fats, naturally occurring saturated fats. So the recommendation is to limit the number of trans fats in food. And as of January 1, 2007, it was required that food manufacturers label foods in the nutrition label with the amount of trans fat that was present. And even before the legislation went into effect, manufacturers were starting to label their food as containing zero trans fat, and they were quite proud of it. The problem is that the legislation has a loophole in it, courtesy of the food manufacturers, because they asked for the loophole. And the loophole is that as long as the food contains less than half a gram of trans fat per serving, the food can be legally labeled as containing zero trans fat. So that means you could actually be eating something that has got trans fat in it and not know it because the manufacturer can legally say that there's none in there as long as it's less than half a gram per serving. So it used to be that a serving was up to the food manufacturer to decide what that looked like. But now there is some uniformity in what is considered to be a serving. And so rather than being a serving by weight, It is now considered to be serving by size of container. So if it says that there is 1 serving in the container, that's it. Or the container represents the serving. And so you now can have a better idea of the number of calories in a container of food, like a potato chip packet from the snack machines, or a bottle of soda from a snack machine, something like that. It gives you the total number of calories per container, not per serving. In the past, there might have been multiple servings in a container, and you would be misled a little bit about how many calories and how many other nutrients you were consuming on the basis of thinking that a serving was equivalent to the container. Now, a serving is the container. But that still doesn't get us around this problem with trans fats. So how can you figure out whether there's trans fat in the food that you're eating or not? You have to look at the ingredients. And if you see something that says partially hydrogenated type of oil, as it says in the label that you can see here in the slide, then you can be absolutely certain that there's trans fat in there, because it is the partial hydrogenation that leads to the formation of the trans fats. And so even though there may be less than 0.5 gram of trans fat in there and it's legally labeled as containing 0 trans fat, if it says partially hydrogenated oil in there, then there's trans fats in there. You can go ahead and eat it if you want to, but you need to be aware of the fact that you are consuming trans fat. So when you look at something like Crisco, which is hydrogenated corn oil, there is a lot of trans fat in there. But the manufacturers of Crisco have never denied that, nor have they hidden it. And they still label the cans of Crisco with how much trans fat is in there, because there's a lot in there, because it is partially hydrogenated corn oil. That's why it's solid at room temperature rather than being liquid at room temperature. And there is this move to get trans fats out of the food industry as much as possible. And there's a lot of effort to try and get the trans fats out as much as possible. Just be aware that if you are eating a food that claims to have no trans fat, that may or may not be correct. Look at the ingredients and decide for yourself whether that is a correct label or not. Be aware of what you're eating, is basically what I'm saying here. There was something else that I wanted to say with regard to fat, and particularly trans fats, but I've forgotten what it is now. If I think of it at a later time, I'll mention it. But I think we're very close to thinking in terms of fats. There's a couple more fats that we need to pay attention to. There is a whole group of lipids that are said to be lipids without fatty acids, and these are built around what is called the cholesterol backbone. These are referred to as steroids. So cholesterol is a type of steroid, but it is in fact the steroid from which all the other steroids in your body are produced. So here is the cholesterol backbone. I'm in red. So there are key features for cholesterol that tell us that it is a lipid. It is pure hydrocarbon, so that's a hydrocarbon tail. This area is all hydrocarbon. The only polar region is this hydroxyl group on the end of the cholesterol. Everything else is hydrophobic. So the hydrocarbon regions are hydrophobic. As we might expect. Excuse me. But out of this, we not only need the cholesterol because it has value in animal cell membranes. We'll talk more about that when we talk about animal cells and the membranes. Cholesterol is a key component of animal cell membranes. But we also create the steroid hormone. So testosterone is a steroid hormone. This is estradiol, which is an example of an estrogen, which is, again, a steroid hormone. Also included in here is cortisol as a steroid hormone. And as you'll see on the next slide, anabolic steroids are also steroid hormones. They are synthetic forms of testosterone, in many cases more effective than testosterone. And they have all sorts of issues when people take, particularly males, but even females, take anabolic steroids. So you have this situation called roid rage that affects males that take anabolic steroids. It leads to high levels of aggression and all sorts of issues, but it also leads to or promotes a couple of different types of cancer that are not particularly pleasant as a consequence of taking the steroids. In the case of a woman who takes anabolic steroids, it suppresses her menstrual periods. It promotes the growth of hair. It rearranges body fat. It changes the voice pitch. It basically starts to masculinize the female body. But likewise, a male who takes too much female hormone or gets exposed to naturally occurring molecules like molecules called phytoestrogens that are found in soybeans that mimic the behavior of female hormones. It can actually feminize a male body. That's how powerful these molecules are. And they are very, very similar. If we go back to the previous slide, you can see that testosterone differs from this type of estrogen by the presence of the hydroxyl group here, whereas there is a carbonyl group, and the presence of a carbonyl group here, where there is a hydroxyl group in the testosterone. Very, very subtle differences. But the effect of these molecules as a consequence of these subtle differences can be huge because of the way they interact with various tissues throughout the body. There's a lot more detail that we could talk about here, but again, in the interest of time, I don't want to go into those details because we're going to be going long enough as it is. So let me just finish up there. So again, another break, that's the lipids and the fats. So again, if you need to go and take a short break, feel free to do so. But let's take a look at these concept questions so that we can move on. Explain the difference between saturated and unsaturated fat. What are trans fats? Human sex hormones are lipids. Why? So what's the difference between a saturated and unsaturated fat? Saturated fats have the maximum number of hydrogens attached to the carbon chain, and the carbons are linked together by single covalent bonds, allowing for the maximum number of carbons to be attached. An unsaturated fat is one where there is at least one double bond in the carbon chain, which reduces the number of hydrogens that can be attached. So it now becomes an unsaturated carbon chain. What are trans fats? Trans fats are a variation on the unsaturated fat form that is found naturally. Remember, the natural form of an unsaturated fat is the cis form. It has a huge kink in the tail. A trans fat is the alternate geometric isomer form, and it re-straightens the carbon chain so that it behaves more like a saturated fat. Why are sex hormones known as lipids or considered to be lipids? Because they are built or derived from cholesterol, which itself is a type of lipid, a very hydrophobic hydrocarbon. So it is a form of lipid as a consequence of that. Okay, this is going to be quite a long recording, unfortunately, but we're getting closer to the end. The next group of polymers to talk about, because the next group are polymers, remember the lipids are not, are the proteins and polypeptides. And proteins make up a significant part of many cells, many plant cells contain a single protein that can account for 40 to 50% of the plant cell protein. That's why this number is so significant. Proteins are really, really important to cells because they are the workhorses of cells. They form structures, they transport things across membranes, they function as enzymes to regulate chemical reactions, they're involved in the immune system to defend us, they are involved in muscle cell structure and allow muscles to contract. They do a boatload of work in cells. So this diagram gives you a short list of some of the things that proteins are responsible for in cells. This is why we need to be aware of proteins as polymers and the importance of proteins to cells. So proteins are built out of a monomer called an amino acid. This is a simple diagram of the basic structure of an amino acid. All amino acids contain this base structure, and this is what leads to the name amino acid, because you've got the amino group sitting here, and you've got the carboxyl group with it, which is an acidic group. This hydrogen actually gets released when the amino acids are dissolved in water, and that's adding hydrogen ions, and by definition that makes it an acid. What makes each amino acid unique is the side chain. So the side chain could be polar, it could be non-polar, it could be acidic, or it could be basic. And each of the side chains and the chemical property of the side chain not only defines what the amino acid is, but determines the chemical property of that amino acid. So leucine, which is one of the two amino acids that are shown here, has a very hydrophobic side chain because it's pure hydrocarbon. So leucine is a non-polar amino acid as a consequence, because its side chain is non-polar. Serine, on the other hand, has a polar side chain. And so as an amino acid, it is also polar. That is its chemical property. So we would expect to find leucine in a region that is non-polar inside the polypeptide. And we would expect to find serine not so much inside the polypeptide, but on the outside of the polypeptide where it would be in contact with water, because it's a polar amino acid. Now, amino acids also form in enantiomers, because as you look at this diagram, you see that there is 1, 2, 3, 4 different things attached to the central carbon. So it's asymmetric, and remember, by definition, that gives us the mirror image isomers. And so amino acids in life are the left-handed mirror image isomers. And if you were, I don't know whether the right word is to say mean enough or sadistic enough, I don't know how, but if you were to feed a rat a diet of right-handed amino acids, which is possible to do in the lab, the rat could eat all of that protein that contained the right-handed amino acids, but it would eventually die because its body would simply not know how to metabolize those amino acids, because they're the wrong isomer. They're the right-handed isomer, not the left-handed isomer. Life makes left-handed isomers and only knows how to use the left-handed isomers of the amino acids. So when we start to make polypeptides and proteins, we are going to start linking the amino acids together in chains. The covalent bonds that are used to link the amino acids together were named peptide bonds by the German chemists who first identified that this nature of proteins. So this is the reason why a long chain of amino acids is referred to as a polypeptide, many peptide bonds. Once those amino acids start linking together, they start to fold the polypeptide chain into very complicated three-dimensional shapes. So there's a sequence of five animations that walk you through, number one, a general introduction to the shape of polypeptides, and then sequentially through the 4 levels of structure that form as a consequence of linking amino acids together to form polypeptide chains. So this is the basic way in which we carry out the reaction. So again, it's a dehydration reaction. So there's our hydroxyl and our hydrogen that we're going to remove. The hydroxyl comes from the carboxyl group. The hydrogen is one of the 2 hydrogens on the amine group. And this covalent bond linking the remnants together of the carboxyl group and the amine group is what is referred to as a peptide bond. If we were to add the next amino acid, we would add it down here. So the next amino acid would be added here. And the chain would elongate in this direction. So that would become our peptide chain. So as I said, the next 5 slides in the presentation contain the 5 animations that walk you through the 4 levels of structural organization that are found or that we recognize in polypeptides and proteins. So this first is an introduction So this is the introduction to protein structure. And again, remember, all of these animations are found in the resource on the resource page for this chapter. So these are there again in their order and identified for what they're covering. This one covers the primary structure. This is called the primary. And it's the sequence of amino acids. So it's a fairly straightforward. This is the simplest one and easiest one to understand. It's just a sequence of amino acids, and this animation walks you through that. But the remaining levels of organization are derived from the sequence of amino acids. So the primary structure gives rise to the secondary, tertiary, and quaternary or quaternary structure, depending on how you want to pronounce it. So this covers the second or secondary level of organization. And this revolves around two structures, one called an alpha helix and a second structure called a beta pleated sheet. So if you watch the animation, it will walk you through these structures. And the key feature here is is the fact that these structures are held together by hydrogen bonds between the amino acids. So they form spontaneously. Maybe I should tidy that up. I was trying to sneak it in and I just did such a lousy job. Tidy that up in there. There we go. Runs off a little bit, but you can read that. So the alpha helix and the beta pleated sheet form spontaneously as a consequence of the amino acid sequence. So there's a consequence of the primary sequence. The key is that the structures are held together by the hydrogen bonds between the amino acids. This gets us into the tertiary level or third level. And this is considered to be the final 3D folding of the polypeptide. Let's clean that up. And there, so it results from interactions between side groups of the amino acids. And so there could be hydrophobic interactions. There could be hydrophobic interactions. We could have hydrogen bonds. Ionic bonds. And in one circumstance, even a covalent bond. And this is all explained in this animation, how this relates. So you can see here alpha helices. So this is an alpha helix. And this is a beta pleated sheet. And this polypeptide, this is another alpha helix. In case you just didn't realize that. But this will walk you through the tertiary level. Then the final level, which is on the next slide, is called the quaternary. Some people say quaternary. My understanding is that the preferred pronunciation here in the United States is quaternary, but I learned quaternary. You can use whichever pronunciation you think is best for you. So this is the fourth, or quaternary. And this is multiple polypeptide. polypeptides form a protein. So a protein, by definition, is something that has function. And in some cases, a folded polypeptide really does have a function. So not only is it a polypeptide, we would now refer to it as a protein because it has a function in the cell. But many proteins are actually formed from multiple polypeptides. So you don't actually have the protein until all the polypeptides come together. And a statement that I teach all of my classes, and I'll pass this one on to you as well, goes something like this to summarize the quaternary or quaternary structure. And it goes like this. All proteins are polypeptides. And that's true. All proteins are polypeptides. But not all polypeptides are proteins. And that's because some proteins are formed from multiple polypeptides. A really good example is hemoglobin, the protein that carries oxygen. It's actually made up of 4 polypeptides, 2 known as alpha polypeptides and 2 known as beta polypeptides or beta units. And you need the combination of 2 of each in a particular form to create the protein that carries oxygen known as hemoglobin. So that's a really good example of a quaternary structure. And there's actually a diagram of that coming up, but I thought I'd throw that in now so that you understood what quaternary is all about. So one of the questions that we constantly have to consider is how can we make so many proteins from just 20 common amino acids, which is the case. There are 20 common amino acids that make up all of the proteins that are found universally throughout life. There are some rare amino acids that have very specific functions, but in terms of universal amino acids, there are just 20 of them. But there are hundreds of thousands of different types of proteins. How do we manage to make them all? Well, just as in any language, you have a limited number of letters to make up the thousands upon thousands upon thousands of different words that make up that language. So you can make up a lot of different polypeptides by simply arranging 20 amino acids into multiple different forms, multiple different sequences. All that matters is that the resulting polypeptide have some function, just as any combination of letters could be a word, so long as everyone agrees to consider it to be a word, that it has a function. That's where new words come from, by combining letters together in unusual ways and saying, well, that's a new word. Why is it a new word? Because we say so. And likewise, a sequence of amino acids becomes a functional polypeptide and a protein because it works. The way that that chain of amino acids folds into its final 3-dimensional shape and perhaps interacts with other polypeptides means that it has function. Why does it have function? Because it does. Not because someone says it does, but just because it does, because of its shape. So I guess its shape says it has function, if you want to put it that way. So we've talked about hemoglobin being formed from 4 polypeptides. Here you can see the normal polypeptide. Some of you may be familiar with a disease called sickle cell disease. In sickle cell disease, one of the 2 polypeptides, the beta polypeptide, has a change in amino acid at position number 6. So it goes from actually it's position number 7. I'm not sure why they've changed this. It is position number 7, and they haven't indicated what the change is, but the amino acid here that is listed as GLU, which stands for glutamic acid, is changed to another amino acid. I hadn't noticed this in the diagram before until I just took a good look at it. So the change is a GLU, which is glutamic acid, to VAL, which is valine. And you're going from an acidic amino acid to a non-polar amino acid. Then you might think, well, a single amino acid, what harm could that do? Well, unfortunately, that particular amino acid happens to be in a very important location that determines how the polypeptide will fold into its final form, and it folds incorrectly, which means that the hemoglobin that results from this, which is called sickle cell hemoglobin, no longer functions properly. It can't carry oxygen properly, nor does it exist as individual hemoglobin molecules anymore. It daisy chains together in long rods of protein, which triggers the red blood cell to change from its normal smooth disc shape to this irregular sickled shape that's characteristic of the disease. And all the issues that result from sickle cell disease can be traced back to this single amino acid change and its effect on the normal folding of the beta polypeptide. What appears to be something of zero consequence actually turns out to have huge consequence because it happens to be in a very, very critical location in the polypeptide. So changing the amino acid sequence can have huge consequences in terms of the normal folding of the protein. So the sequence of amino acids really is the source of all the other levels of structure in the protein, and messing with the sequence of amino acids can have huge consequences, as we can see here. Misfolding of proteins is also associated with diseases. The infectious particles known as proteins, as prions, are simply misfolded proteins. And what is really scary about these is the prions cannot be destroyed by cooking. Now, why that's important is that we know that proteins are heat sensitive. So when you cook an egg, the transparent region that we often refer to as the white is actually a concentrated solution of a protein called albumin. And when you heat the egg, when you cook it, the albumin loses its shape, a process called denaturation. And it changes the solution from a transparent liquid solution to a solid opaque material because of the loss of shape of the albumin. And that's irreversible. You can't uncook an egg. And it turns out that proteins are very sensitive to environmental conditions like pH, hydrogen ion concentration, temperature, presence of different chemicals in the environment. But some proteins, for reasons which we don't understand, and this is the case with the prions, have taken on this misfolded shape. We don't know the origin of the original misfolded prions, but what we do know is that when those prions turn up in the presence of normally folded proteins, they trigger the normally folded proteins to misfold. So the prions can trigger normally folded proteins to misfold, which causes disease. all of which are incurable and 100% fatal. So what sort of diseases do we know of that are caused by these prions? This is where it starts to get a little scary. So you've heard of mad cow disease, otherwise known as bovine spongiform encephalopathy, by saying that really quickly. Bit difficult. So mad cow disease is caused by prions. The human form which you develop by eating meat that's contaminated with the BSC prions, is called Creutzfeldt-Jakob disease. So let me move the text box. So it's called Creutzfeldt-Jakob. It's actually with, okay, so let's take that out. Jakob disease. And this is why people are concerned about importing meat, and particularly beef, from other countries, and particularly from Europe, where the only known human transmission of mad cow disease to humans has actually occurred in Britain, and at least 3 people have died of mad cow disease since it was first recognized as a consequence of eating contaminated beef. And because cooking can't destroy these prions, it doesn't matter how well you cook your hamburgers or whatever it is that you're cooking. You can't destroy the prions. They're already misfolded, so you can't misfold something that's already misfolded. And the mere presence of these prions causes other, the normally folded proteins to misfold to match the prions, and that's what gives rise to the disease. Another condition that was found primarily in New Guinea amongst the cannibals of New Guinea is a condition called kuru, and you can see the spelling here again. The transcription service is struggling with this, but this was passed from generation to generation because amongst the cannibals, it was considered to be a sign of respect to eat the brain of your dead relative. And of course, if the relative died of kuru because of the presence of the prions, eating the brain, whether you cooked it or not, would simply pass on the prions, and eventually you would develop kuru and die of that, and then your relatives would eat your brain, and it would get passed on from generation to generation. And it took a lot of time to convince the cannibals, in which eating the brain of dead relatives was a cultural tradition, to stop doing it. I mean, cannibalism is pretty much unknown in New Guinea now, but it was still known into the 1960s and early 1970s. And eating the brains of dead relatives went on past the classic period of cannibalism. But my understanding is that they've more or less eliminated this now because they've been able to help everybody understand what the nature of kuru is and how it is passed from one generation to the next. And so they've come up with other ways of honoring the dead relatives other than eating their brains. Let me move the box back over here again. So there is another condition called scabies. which is found in sheep, and this is again another prion disease that affects grazing, sheep as grazing animals, but it also seems to affect deer and elk and moose, so antelopes, which are closely related to sheep. So this is another one of these prion-caused diseases. And again, eating contaminated meat is all that is necessary. So you can understand why these diseases are really, really scary. You can't kill off the proteins. You can't destroy the proteins by cooking them. And all you need to do is eat some contaminated meat that contains these misfolded proteins, and it will affect the proteins in your brain, and eventually you will develop the condition, whether it be mad cow disease or the human form, Creutzfeldt-Jakob, kuru, if you were game enough to eat a dead relative's brain that died of kuru, or eating contaminated wild game or sheep that have scabies. And you can understand also why when a number of years ago, several cattle herds were in the United States were identified as having members with mad cow disease. They simply slaughtered all the animals on the farm, plus the surrounding farms, and then put the bodies in big pits, soaked them in gasoline and set them on fire to destroy as much as possible, and then buried them in the hope that eventually the proteins would degrade, because you could not allow them to come into contact with any animals that would be in the human food chain. It was just too risky, because it's just coming in contact with these misfolded proteins. That's all it takes. Scary diseases. And then I mentioned earlier the types of things that will cause proteins to so the loss of shape. So this is what happens to the proteins of an egg when you cook it. So, and in fact, if you want to sacrifice an egg, you can see that pH affects this by cracking an egg into a glass and then adding some lemon juice. And you'll see the same effect. The white will turn white in response to the presence of the lemon juice, because you've altered the pH. And all you've done is denatured the albumin in the egg white. So that's proteins. That's the third of the 4 macromolecules. We're getting close to the end, I promise. So one more break period. Actually, we've got 2 break. We've got this break period, and we've got one at the end of the nucleic acids. And then I'm trying to remember whether I put one at the end after we talked about lactase persistence. I don't think I did, but we'll see. Anyway, concept check at this point. How can changing a single amino acid in a protein change the function of the protein? Give you a second to think about that. So if you immediately thought about the change in amino acid that gave rise to the change in hemoglobin that leads to sickle cell, you're on the right track. Because if the amino acid is in a key point in the polypeptide, it is going to change the way that polypeptide folds, which almost certainly will change the function of the protein. Because remember, The function of a protein is a result of its shape. If you change the shape in any way, you're obviously going to have an effect on its function. So the final polymer to talk about is the nucleic acids. There are two major nucleic acids that we need to be aware of that are polymers, and that's DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA, of course, we know is the molecule of inheritance. It's where the genes are located. It's the constituent molecule of chromosomes that contain the genes. And it's that information that is passed on from one generation of cells to the next and one generation of organisms to the next. So when we think in terms of a gene, all we need to realize is that a gene is a unit of coded information within the DNA molecule that typically codes for the amino acid sequence of a polypeptide. And so when we think in terms of cellular information, we think in terms of two languages: the language of nucleic acids, of DNA and RNA, and the language of proteins, the language of amino acids. And when we talk about extraction of genetic information and utilization of that information, we talk about two processes called transcription and translation, which are language terms. Transcription means changing from one form of language to a second form of language, such as from written to spoken or from spoken to written. And translation, of course, is changing from one language to another, so from English to Spanish or Spanish to English. And those are pretty good descriptions of the processes that involve DNA and RNA in terms of cellular information processing. But we'll talk more about that in chapters 9 and 10, specifically chapter 10, in fact. We'll talk about transcription and translation in more detail. But we do recognize this flow of information. This is sometimes referred to as the dogma of molecular biology, but the flow of information is from DNA to RNA, which is a change in the form of language, both being languages of nucleic acids. So this would be transcription. We might as well put these in here, even though we're not going to talk about them in detail. And then when we go from nucleic acids to amino acids and proteins, this is translation. In fact, you'll be creating the second of the two infographics that you're going to be producing this semester to talk about transcription and translation and also DNA replication, but that's more on that later this semester when we get to that point in time. So the nucleic acids are polymers, and so the polymers are called polynucleotides. So the polymers are called polynucleotides because they are strings of nucleotides, and the nucleotide has three components: a sugar, and the type of sugar determines whether it's DNA or RNA. So in DNA, it's deoxyribose. In RNA, the sugar is ribose. So the actual name of the two nucleic acids tells you that is built around the sugar. So DNA is deoxyribose nucleic acid, and RNA is ribose nucleic acid. So we have the sugar. Attached to the sugar in what is referred to as the 5' location is a phosphate group. And so all nucleotides also have a phosphate group. So the sugar and the phosphate are the common features of every nucleotide. What makes the difference is what is referred to as the nitrogenous base. So in DNA, the 4 nitrogenous bases are given the letter designations A, G, C, and T. And because they are what create the difference between the 4 nucleotides, we also use those letter designations for the 4 nucleotides. So A is adenine, G is guanine, C is cytosine, and of course T is thymine, as it's suggested here. So those are the 3 components of a nucleotide. And the same is true in RNA. Each nucleotide has the sugar ribose with a phosphate group attached to it, and then 4 nitrogenous bases. But in RNA, uracil replaces thymine. And they are almost identical. There's a very subtle difference between these 2 nitrogenous bases, but we substitute uracil for thymine in RNA. So we've actually seen 2 of the 3 differences between DNA and RNA. Different sugars and a difference in the nitrogenous bases that are found in the nucleotides. So I've already identified this, but we'll keep moving on. So here are the 4 nitrogenous bases, and there is a complementation. We say that the nucleotides complement each other. Complement, or another term for it is base pair. And so A pairs to T and C pairs to G. So these 2 form a pair and these 2 form a pair. And that's an important understanding of the nature of the DNA molecule. It also tells us something about how we can copy the DNA molecule. There's a lot involved in terms of understanding the function of the DNA molecule by understanding a lot about its structure. But that shouldn't come as a surprise because we know structure and function are correlated. So no surprise here. So again, it's a dehydration reaction, nothing new there. So when we link them together via, and I was hoping I had a diagram in here, but I don't have a diagram in here that shows it. Maybe it's a little bit later in terms of that. So when we think in terms of the DNA molecule, yes, a string of nucleotides is called a polynucleotide, but a DNA molecule is a double-stranded polynucleotide. So there are 2 strings of polynucleotides, and they are linked together. A pairs with T, G pairs with C. When DNA is copied, so Watson and Crick actually first identified, and Watson and Crick, by the way, in case you don't know, are the two gentlemen who published the first description of what the DNA molecule looked like. In that paper, Watson and Crick actually suggested how DNA would be copied. It was pretty obvious to them looking at their model how the DNA could be copied, and they called it semi-conservative, meaning the two existing strands of the DNA molecule were conserved and they functioned as templates to form the missing strands using the base pairing rules. And it would only be 3 years later that a paper was published that confirmed that yes, DNA is in fact copied in a semi-conservative fashion. And again, we'll learn more about that when we do chapters 9 and 10 later in the semester. But you can see the basic structure here, the famous double helix model of the DNA molecule. There is something else that we probably should put in here. One of the things that we recognize is that there is a directionality to the strand. So one end of the strand is going to be referred to as the 5' end, and the other end of the strand is referred to as the 3' end. And I've already indicated that this numbering exists on a previous diagram, but let's relate this to the DNA molecule itself. So if we look at one end of one of the strands, it might have its 5' end up here. If we follow this all the way through, of course, we expect to find the 3' end down here. Well, what we then notice is that the complementary strand doesn't have its 3' end here, but its 5' end, and the 3' end is actually back up here again. So that would be 3' end. And we say that the two strands are not parallel, but anti-parallel. So they're anti-parallel. And we can illustrate this by saying that one strand is going 3' to 5', like that. The complementary strand will be 3' to 5' in the opposite direction. That's what we mean by anti-parallel. If they're parallel, they both go 3' to 5' in the same direction. So this slide is just reinforcing again the similarity between DNA and RNA in terms of the monomers. But of course, we do know that there are differences. So we've looked at two of them already, the difference in sugar, the difference in nucleotides. We've already mentioned those two. Here's the third one, that RNA is considered to be a single-stranded polynucleotide. Whereas, as we've seen, DNA has two polynucleotide strands. So that's the third difference between DNA and RNA. So this time we're looking at RNA nucleotides in this diagram. Because we've got the ribose, we've got the phosphate group, and again, this is the 5' sugar. Where's the 3'? I didn't label it before, but this is 3' here. So this is 1, 2, 3', 4', 5'. Why is it given the name? Why is the prime designation given here? Well, if you look at the nitrogen spaces, you can see that there are carbons there as well. And so the chemists who first deciphered all of this decided that in order to be able to differentiate between the carbons of the sugar versus the carbons of the nitrogenous base, the sugars would be given the prime designation. So in ribose and deoxyribose, it's 1', 2', 3', 4', 5'. but it's the 3' and the 5' that are important in terms of the directionality of the polynucleotide, because the link is between the phosphate on the 5' and the hydroxyl group on the 3'. So the next nucleotide in terms of its phosphate group would be found here. Oops, that's not what I want. So its phosphate group would be here, and this would be to the 5' carbon of the next nucleotide, and so on. And so that's why the 3' and 5' become important. Again, we'll learn much more about that when we do the DNA chapters later in the semester, chapters 9 and 10. So, final concept check. We are almost there. Seems like we've been going forever. So there are how many strands of polynucleotides in a DNA composed of how many nucleotides? If one strand in a DNA molecule contains this sequence, the complementary strand would have what sequence? And as I said, time for a break and a cheesy joke. Yeah, this one's about as cheesy as they get. Complete this biologist pickup line. Of all the nucleic acids, RNA is my favorite because it has blank in it. Give you a few seconds to think this one through. Okay, so in DNA, there are 2 strands of polynucleotides composed of 4 nucleotides. So if a strand had this sequence, 5' G, A, T, G, C, 3', then the complementary strand would go 3', C, T, T, A, C, G, 5'. So that would be the resulting sequence. So we would put in here 3', C, T, T, A, C, G, 5'. And now for the cheesy joke. I promise you it is really, really cheesy. It is the most pathetic joke. Of all the nucleic acids, RNA is my favorite because it has U in it. I told you it's cheesy. Anyway, on we go. So just coming back to look at what gives rise to lactose or lactase persistence. Genetic analysis, study of the genes has indicated that when you look at chromosome 2, where the lactase gene is positioned, if you have one nucleotide, and I believe it's the C nucleotide, you have lactase persistence. If, however, you have the T nucleotide, you will lose the ability to make the lactase enzyme. That's the change. And notice it's not actually in the gene itself, it's in another location. So these nucleotides And we are actually going to look at the regulation of genes in eukaryotes like ourselves. So we'll be able to talk a little bit more about why this control region exists and why it can exert its influence over the lactase gene to either determine either you produce the lactase enzyme or you don't produce the lactase enzyme, depending on what nucleotide is found in this control region. So if you happen to be, well, it depends on how you want to look at it, fortunate enough to continue to produce lactase and you can enjoy milk and dairy to your heart's content, now you know why you can do that. Should you really be able to do that? Well, as a mammal, no. But as a human, because of our cultural traditions and the use of herding in some cultures, that has favored the appearance of this mutation, which has meant that for those of us who are lactase persistent, we can enjoy dairy products. For those of you who unfortunately do not have that mutation because either you didn't get it or you come from a culture where herding is not a tradition, unfortunately, you can't really enjoy dairy products. Although there are dairy products now that have had the lactose removed from them so that you can enjoy milk and other dairy products as well, or you can buy lactase enzyme in pill form, in capsule form, so that you can swallow that before enjoying dairy to get around the problem of lactose intolerance for those of you who happen to have lactose intolerance in its many forms. So now you know. We've come back and we've addressed this, and you can see what the deal is here. So for individuals that come from cultures where there is not a strong herding tradition, lactose intolerance has a very, very, very high percentage of lactose persistence, very low. But in cultures where there is a high tradition of herding and the use of milk as a food source, there is a very low level of lactose intolerance and a very high level of lactose persistence. As I said, on average, in any population, about 70% of people have some form of lactose intolerance. 30% of the population have lactose persistence and can enjoy dairy without problems. So now you know. So the last couple of slides are just information slides summarizing the different types of macromolecules and some features that may help you start to put together the pieces. I do realize, unfortunately, that this has been quite a long presentation. I don't know yet exactly how long, but I know it's at least several hours long, perhaps longer than that. Maybe close to 3 hours long, maybe even longer than 3 hours. But it is very information rich. What I would encourage you to do if you're coming back to take a look at this, and I'm going to send out an email when I post this presentation a little bit later today, break it up into units. Don't try and watch it all at once. You get into information overload really, really quickly because there is a lot of information in this. It is unfortunately a product of the topic. We cover a lot of material. What we've covered in this one presentation, in my majors class, we cover in at least 2. Potentially we could do it in 3. So there's a lot of information in here. I hope that you found it interesting, that it's relevant to your life, because it's talking about those molecules that we call food molecules. And it's talking a little bit about the importance of those molecules and why they're important to us, and also something with respect to how we consume those food molecules and what impact that could have. Anyway, if you have any comments, concerns, or questions about anything that's been covered in this presentation or any of the 3 presentations so far, by all means, come and see me in office hours or shoot me an email, whatever mode or method suits you, and let's clarify whatever it is that you're having issues with. But I will see you again next week for the presentation on Chapter 4, which is all about cells, membranes, and cell constituents. So until then, bye for now.